good afternoon all. So hope I am audible. Just anyone please confirm.
So yeah, so let us start. So I'll be starting with the presentation. So just let me know if my screen is visible. Yes, okay. Till now, okay, fine. So I'll be starting with the live session. So welcome you all in the week 2 of the live session. So here we will be starting with the week two's contents, that how what are the different contents and different problems, and also as we have discussed in the previous session, that we will go with uh, one of the case study. So did any of one of you tried what I told about the SRA toolkit? Had any one of you tried looking or studying about that? Yeah, anyone?
Yes, sir.
One second, would we? I am not able to hear you. One second, let me check. Yeah, would be. So you were not able to download it, right? SRA toolkit you were not able to download. Okay. Any other one? Anything else? Okay. So if you haven't tried, sorry. Yeah. So Udvit wrote in chat that she did not able to download it. Anyone else tried for the SRA toolkit? No one. Okay. So Udvi, you tried using which system SRA toolkit to download? Udvi, I am audible, right? Windows. So actually, these are tools are used in the Linux based tools. So the thing is that it would be better if you can use some virtual machine or VM. You can go and search about what is a virtual machine. So using virtual machine, you can try using that. Okay, in a Linux based platform. Then it will you will be able to extract and start installing and working it. Okay, one day I will one day I will show the steps and the different tools in our PC. So you try to know about the virtual machine, because analysis of all this requires a little bit of knowledge in the Linux, this virtual machine, in the different packages in the Linux based platform. So then only you will be able to use it. I don't know why I not able to hear you all. So fine. So let us start with the today's session. Here today's case study with the same paper of the proteogenomic characterization of human colon and rectal cancer. The previous day, what we uh, what we have seen of the correlation between the different mRNA and the protein level, that how the single amino acid variants effect of the single amino acid variants at the different levels of the protein levels. So now today we will look at the different copy number alterations and its effect on different its effect on different kind of at the protein level or the mRNA level. Okay, so why, before we go and see that the with the gene genomic sequence, and we can analyze our data in such different things, in such different formats, and the most importantly you all will appreciate is that these simple gene level changes and this level changes, how important is to understand and read their studies, as you all know that it is causing so many different types of diseases, and these cancers are effect of this level of this because of all these little small alterations at the genomic level. So here if we see that what is the copy number variation in and its correlation at the mRNA level, or what is the correlation uh, at the protein level. So you all are able to if you all cannot see my slide or anything, or cannot hear, just let me know. So yeah, so the so the copy number alteration. So in this paper what they have done, so see here is the gene location in the y-axis, if you can see all. So I will suggest you all to go and read this paper, at least start with this paper, because you will get an understanding that how this analysis, what analysis is done after the sequencing technologies. So here you can see the gene location in that chromosome wise from 1 to 22 and the XY chromosomes, and which location which gene is the copy number is there, and how it is correlated with the at the mRNA level and at the protein level. So here you will get to know that whatever is coming is correlating. All the effects are not coming at the protein level. Okay, so there is correlation at the mRNA level, positive correlation, but similarly if there it is there at the mRNA level, it is not getting at the protein level. So here different copy number abundance is been used using significant positive and p value less than 0.01. It has been tried to understand that how it is correlated. So if so there was a I will not say myth, but a kind of myth that the cancer is only the cause of the genomic changes, and if there is a mutation or if there is a variation, then the difference occurs and it is responsible for cancer. But this has been broken. If you go and check at the other level, like the mRNA level or the protein level, you can then realize and the fact that it is not always the mutation, its effect, how it is being correlated, that plays a major role. And there here comes the importance of the proteogenomics approach to understand and to understand the at the whole molecular level that what changes are happening and how this is happening. So here if you go and understand that impact of the copy number alterations in this paragraph, you will can identify that the TCGA has identified 17 regions of focal amplification, 28 regions of significant focal deletion. So somewhere there is an amplification and somewhere there has been a deletion. So now they had examined this impact of CNS on mRNA and protein abundance, and including both the cis effects and the trans effects. So I think you all know that the cis effects is that when the effect is on the same DNA of the gene, and in the trans is on that uh, at the different chromosome where the effect is seen. So all the 23,125 genes with the CNA that has been measured in the TCGS dataset, they have performed a peer correlation. Also, one thing you can see that all these approaches, all these techniques, all these things are correlated in a mathematically using only Spearman's correlation, and all those statistical tests are very much important to understand to interpret the biological data, why it is happening, how these things are happening. So this is at the 3764 genes with both mRNA and protein measurements. So that another drawback still lies is that all the genes we can examine at the genomic sequencing by sequencing at DNA and RNA-seq, but all those proteins cannot be detected in the mass spec base due to the different limitations of the abundance and all. But still those of the genes that are getting uh, expressed and getting understood at both the levels. So now, so examination of this matrix, and here using strong positive correlations, so which are above the diagonal, along the diagonal they will have the positive, strongest positive correlation. So they suggest that strong cis effects of CNS on mRNA abundance. So the cis effect is stronger, but previously diagonal pattern was much weaker. So here you need to focus, and here is the major thing, that the diagonal pattern was much weaker for the CNA protein correlations. So here you can see that at the mRNA level it has much more effect, that it is showing much more positive correlation, but at the protein level it is not so much. Means that CNA, if it is also affecting at the mRNA level, all are not getting translated at the protein level. So the changes that are happening, it's not that always what copy number alterations is getting seen at the protein level. So what we generally are thinking will be that there is a change in the genetic level, hence there will be a change in the protein level, or there is an increase in abundance or the differentially regulated. But here we can see that this trend can be altered also. That means that from mRNA to protein level there can be some changes which is not allowing it to correlate positively at the protein level. So another is that to investigate further the cis effects of CNS, so they separated all genes with CNA, mRNA and protein measurements into those in focal amplification regions. So I am just pointing out these paragraphs and these two because we want to understand that how to that experiments, that the data, this is not simply that ATGC sequence or simple protein amino acid sequence, but the way the data can be looked upon, the way the data can be seen, the way the data can be analyzed, and the way we can derive on the conclusions on why this on what are the major driving forces behind occurrence of such genes. So here, after this correlation, they did not stop, and they tried to separate all genes and focal amplification regions, their deletion regions, and the non-focal regions. So they design these experiments, and they plan in this way, the analysis is planned in this way, so that all the information that is being there, we can extract it out and can identify and can work on it. And non-focal regions, so now the CNA mRNA correlations were significantly higher than CNA for all genes in 3 groups. So all in this amplification region, be it in amplification region, be it in deletion region, be it in the non-focal regions where no amplification or no copy number alteration variation is there. So all where they are significantly higher in CNA mRNA correlation, but CNA protein correlations were not there. So I would also suggest you all to think that what can be the reason for this, why CNA mRNA correlation is not getting reflected in the protein level. So here again a statistical Kolmogorov-Smirnov test is done to identify that if these changes are significant or not. So here you can see, so the same trait was not observed in the genes in the focal deletion regions. So therefore it has been seen that the focal amplification regions, where the regions are amplified, have the strongest cis effects on both mRNA and protein, suggests that selection for high protein abundance may drive CNA in regions of focal amplification. So what they concluded is that, suggesting that selection for high protein abundance may drive copy number alterations in regions of focal amplification. But this is their interpretation, their way of thinking. So you all can also think, go and read and understood and try to interpret it in a can interpret it in a different way as well. So however, many CNA driven mRNA do not translate into increased abundance of the corresponding proteins. So there it has been evidently found out that many CNA driven, which increases the mRNA level, is not get translating into the increased abundance of the corresponding proteins. So this is a very crucial observation, and this also shows that how the integration of different omics level approaches, or the integration of the different techniques, can help us in getting the information of those, how that it is not corresponding, and to let us think and explore much more to understand at the molecular level how we can go and identify this. So this is so I thought to give a case study with that paper only. So we will complete that paper other, and also we will go for case study for all this, and also I'll suggest you to go through these ones, because here you can get an idea about how much. So if as is anyone has any question, so can ask or write in chat. I don't know, there is some issue in the audio thing. So does anyone have any question, or everyone is clear of what? Also I thought of showing you the CNS, because we will be doing in the later stages the CBio portal, where we will be looking at the data at the different variations and all. So focal amplification region is that region where the copy number in the particular sequence is gets multiplied. So whatever is the required number, it gets altered and it gets multiple times it is there. That is where the amplification happens, and where somewhere it is deletion, so then the copy number gets deleted. That is the deletion region they tried to mention about it. So by looking at the, when you will see that there are various data, various types of variants, structural variants, copy number variations, copy number alterations, but what do they implement, what do they actually mean to? So that becomes important. That becomes that okay, so this is the so this can be, by looking at those data you can rethink that okay, so in this data this is the copy number, this is at the mRNA level, this is at the protein level, so what can be the changes that could have happened. So this will encourage you to understand that why this studying this variance, looking at this data is so much important carefully. So okay, so if so Kolmogorov-Smirnov test is a statistical test parameter, like you, I think you all know so about the students t test or chi-square test. So Kolmogorov-Smirnov test is another kind of statistical test. You can know about where the significance is known. So if you want to study more, so you can go and study. So this test, because this is a, it is a probability based test, so here we cannot describe the test fully, because that then it will become like a statistical thing, so many things are to there. So I can just give you a brief idea, like it's a test to find the significance, like the t-test, chi-square test, what you all are familiar with. So here also it helps in the probability, on the basis of the probability and the it tries to identify the significance, so that you can go and study about it more later. So just for now you let know that it is a kind of a statistical test, uh, that you can study and can know the full equation and the principle of those tests, and then you can perform it. So if CNV and protein level is not may, it is not the reason behind cancer. No, this thing can also not be told, because you can know that there are many proteins which induces apoptosis. So if you go and look at the cell cycle and cell regulation, so there are many proteins which is required for the apoptosis. Now these proteins also, they are co-regulated, they might be co-regulated with someone, or they might be what you say that one protein induces the function of another protein. Now for instance, this copy number variation, for some what reason, the protein which helps in inducing apoptosis, it might be a, I'm saying that it might be that the protein which is inducing apoptosis, if that protein's expression gets reduced, then the cells will not undergo apoptosis at normal stage, and it can affect the cancer. It can help, means it will not undergo apoptosis. So we cannot say in this way. So the CNVs has definitely a role. So if you, because they changes, if there is an alternate CNVs, definitely has a role, but how, why it is not have showing the correlation at the protein level abundances, that's remains an interesting part. Okay, so now let us uh, look. So now another thing I think you all have gone through in this lecture about the expression quantitative trait. I thought to just give a briefly tell you that QTL is a nothing but is a fraction of the genetic variance on a gene expression phenotype. So the genetic variance that has been told at the gene level. So what is this changes at the gene expression in the phenotype? So standard QTL analysis. So I just wanted to focus on this. So involves a direct association test between markers of genetic variation. So markers which shows that there is a genetic variation, and with the gene expression levels. So this is just a test. Again, this is a correlation of the statistical test that just shows that how it is being affected, and it is calculated that it is significant or not, and is typically measured in tens or hundreds of individuals. So I think you all have gone through, but I thought of just showing that that this is just a direct association test, a statistical test, and nothing else, for identify that how much genetic variation is giving at the gene expression levels. So now another thing is that I wanted to share you that this uh, there are, I told that this EQTL requires your different statistical tests. So one statistical test, Quotient Armitage test, trend test. So it is also used for identifying this EQTL, but in a different manner than Pearson test. Okay, so Pearson test correlation, what is used? It uses this test uses a little different way. So power is often improved as long as the probability of having disease increases with the number of disease associated alleles. So anything, it if the probability increases, that if an allele is expected to be associated with disease, so the probability of having disease will also increase. So if there is a change in the disease associated in an important allele, then you can say that yes, the probability is increased. So this is a general phenomenon. So here what they take is that they do an additive version of this additive model. In the additive model they take, so for example, here if you see are counts of the genotype cases, in counts of the genotypes in controls and N0 case control study. In the cases means here in the disease condition, M0 M12 in controls and in the case control samples. So then you can find out that this is the equation with which this test, by adding all those sorry, in an additive model they perform this test. So the different statistical models are used to identify different in a way to correlate and to identify those tests. So here this test uses an additive model to identify the different significance of the variation of the genetic at the phenotype level. Okay, so I just wanted to give a brief description of this EQTL and one of the type of the test, so that you can identify. Also, you all can go and read about different statistical models that are used for the quantitative trait loci analysis. Some might use the additive model, some might use the different other models. So what are those models and how are these approaches? But you can see that this statistical test and statistical uh, plays an important role in understanding and analyzing in all those types of data. So since we cannot explain everything here because we have a short time of, so I'm just giving you all the things that you can go ahead and study more and understand more, and also explain. So any question on in this? Uh, so the its importance is that see the see no model is so no model. There are many statistical models like Pearson's correlation, Spearman's correlation. Everything will give you a correlation, everything, right? But every model or every algorithm, every statistical test has its own pros and cons. So, for instance, previously they used Kolmogorov-Smirnov test, but they didn't use students t-test. Why? Because there are some every these statistical tests are based on assumptions, probability, some assumptions every test does. So every test taking their assumption is different. So in every answer every test cannot give. So that's why people try to identify different types of tests, build new statistical tests, build new models, so to overcome the limitations of the previously used models and tests. So there are different tests available. So I just wanted this test. So this test after forming also they gave an enhanced version. So why they gave an enhanced version? Because to overcome the limitations and to identify uh, all those that this here for the genome GWS studies. So what are the these tests? What are its p-value? Means what are the advantages? What are the disadvantages? So if you go and study, check this paper out, so you can understand more about it. That how it has calculated the p values at the different conditions, so how much it is effective. So each statistical model is how much effective in identifying or in answering our questions. Okay, so here generally what I to focus is that they use an additive model where they take into account of all those counts of the genotypes, and then try to identify that there is a uh, what is the what is that uh, what you call the correlation between the genetic variance and this gene expression. So this is what I wanted you all to emphasize and understand. Okay, so now we so now so here this is the cbio portal. So here we will go step by step, we'll look at the data. So I wanted to give you the firstly the this paper, this was published, this paper, in this paper is that publication paper of the cbio portal. So what they have said that the cbio cancer genomics portal, so open access resource for interactive exploration of multidimensional cancer genomics datasets. So they just think that why these datasets or these free available datasets are important, because for example you're looking at a way of data, my looking at a way of data, or any other person way of looking data is different. So if we perform one experiment and if we generate some data, and if we can we analyze it in our own way, but if we give it to the freely accessible to all so there different people can look at the data in different way, and they can give an different viewpoint, can conclude differently, and we can get new knowledge. Okay, so so another thing is that this all this analysis is very much computer extensive, very much knowledge you need to have on the command lines. Okay, so all these tool, if you see about this genomics data analysis, requires a linux platform, command lines, different hurdles are there. But so all the researchers, but you can, because of these hurdles there will be a limitation to understand and interpret those data. So but it is might be one can give another viewpoint. So between complex genomics data and for instance you just think that there is a huge raw data generated in the form of 80 gc, so how you can go ahead and try to decipher those. So this is helps in bridging the barrier. Complex genomic data and cancer researchers think high quality access to molecular profiles and also the clinical attributes. So clinical attributes is also now researchers across the worldwide are appreciating that clinical attributes are required to understand that how, what are the clinical parameters of those patients, to understand and correlate those data in that way and to understand. So from large scale cancer genomic projects, so and empower researchers to translate into a biological insights and clinical applications. So this is the major important thing. So you all can go and read about this paper that understand so why these open platforms are important and how they are helping us and how they will help us in further going analysis. So today now we will go stepwise so that you can understand some data. We can look at the data. Together we can look at this portal. Look, try to understand the different data type, different data that analysis that have been there, and then try to look at that how we can interpret, can we interpret things differently, or what we can interpret. Okay, so I want everyone to please go to CBio portal and do it side by side. So we will go step by step. So I'll also, okay, so please everyone go to CBio portal in the link. So if you all want, I can paste in the chat box also. Just let me know when you all have opened to the CBio portal. Just write something okay or yes, you have opened. Then I'll proceed to the next step. Okay, Chitra, yes. Thing is that yes, so these models are used for correlational studies. So only Chitra is done. Anyone and other people, please tell. Okay, so many have so all of you have opened, I guess. So we will start. So you can everyone can see this portal where there is a pan cancer studies. All the things you can see that it's a huge page that I've opened, so you can understand the volume of the data that is being already done and analyzed and has been put up. But still, we need to reanalyze, we need to rethink, re-understand the molecular level at the changes at the molecular level of these diseases, and try to interpret and give try to help try to give novel insights on these diseases, right? So now is that next step what we will do is that, so we will filter it out for breast cancers. Okay, so we will go and filter it out for breast cancers. So how many studies you can see here for the breast cancer? So write in the chat box. Yeah, everyone, let us make it interactive so that we can try. Let us see that if we can interpret something else uh, or something, we can give some insight or not, we can gain some insight or not. Yes, only three. Rest of you, please participate. uh, it becomes, yeah, okay. So breast cancer, here we can see that it 24 data sets. So now if we go, so we all select all studies. Okay, so here you can see that breast cancer MSK cancer cell publication, how many samples? 1918 samples. Breast cancer MSK nature cancer 20, how many samples? 141 samples. So you can go and individually also check, or you can select all listed matching studies. So here we are going to do select all matching studies. We are not looking at those. Okay, so you all have selected it, right? All those studies. So you can see that you can filter it out based on your study. So for example, you have reading one paper and want to check that what they have reported, and if you can look those data at the different level, you can go and study. Similarly, you can select all those studies. Now we will, what we will do is that next step is that, so here we have got that answer that how many studies are there? 24 studies. And in 24 studies, individual studies, there are number of samples are also mentioned. So we will go for these three genes. So you'll know that these are some important genes. So we will first go and do submit by query by gene. Okay, so here this can be studied and explored by two ways. We can select one study and can explore, or we know particular gene, or we have targets in mind by literature survey. We know those genes and how those genes behave in all those data sets that are available. We can go and look. So we will do this thing. Okay, so we have done uh, query by gene. So everyone, please respond that you have gone till this place or not. Just write yes or something fast. Everyone, yeah. Okay, fine, fine. Now see what are the, we have need to select the molecular profile. So these genes BRCA1, first we'll write these three genes: BRCA1, BRCA2 and IDH1. So here I will just let you know that BRCA1, BRCA2 is the known mutations for the breast cancer, and IDH1 mutation is known in a glioma, is mutations is known in gliomas in a brain tumor. So now we are curious that how a mutational marker that is known for brain tumor, glioma, how they are coming in the breast cancer. So just think that why, how we can think of these studies, that this marker how it behaves in the breast cancer. Similarly, we can look for this BRCA1, BRCA2, BRCA1, BRCA2, how they behaves in the brain tumors. So in this way you can understand. So now if you see that this IDH1 level is how much this their mutational status, how, what is their structural variance, what is their copy number alterations in this breast cancer, which is not known as an IDH1 mutant. So here we can look and try to identify some, understand something. So for example, if someone is working in pancreatic cancer, can look at for the what are the expression levels of the brain tumor markers, or vice versa. So that's why. So everyone, have you written right about this? So you all clear? So we have chosen these three genes. One is the BRCA1, BRCA2, which is known for the breast cancer, and one IDH1. Okay, so now we will go. So this we will clip it, and then we will submit our query. So all gene symbols are valid. So now we will submit our query. Okay, so see, so now this tab has come with the studies of origin samples profiled for mutation. So this is BRCA1, this is BRCA2, this is IDH1. So genetic alterations. So study of origin means the this color coding is giving for the different kinds of studies, the 24 studies that we have chosen. This is the study of origin, and the genetic alteration here is that inflame mutation, which is a putative driver mutation. Inflame mutation of unknown significance. So driver mutations are known that leads to the disease. Missense mutation again is a driver mutation. Again, missense mutation can be of unknown significance as well. Splice mutation, truncating mutation, structural variant, germline mutation. So there are mutations that come at the germline level as well. So there also you need to understand that you want to check the germline variant. So if you have a genomic sequence and align with the reference genome like HG19 or HG38, then you will get both germline and somatic variants. Now if you want only the somatic variants, then what you do is that you take the genomic level DNA from the uh, tumor region, and the same from the same patient PBMC or non-tumor region area the DNA, and align it to identify only the somatic variants. Then the amplification, deletion, and no deletions. Okay, so this you, I think you all have got it. Now find out. So what we will find out is that we will go at the plots. Okay, so you all can go and like cancer type summary. So I am showing all the tabs one by one, that what happens. So this is by study type. Okay, we are queried genes, we are choosing the BRCA ones. I'm just showing you that how you can go and click and try to know all those data, what are those present, and try to identify. So here we can select cancer study. Let us see, let us go one by one. So if we take out cancer study, so y-axis value is the alteration frequency or absolute counts. Means absolute counts means at the absolute level, or the alteration frequency we can give, so we can get a better idea. And then y-axis values, or alphabetically of the studies we can change it. So minimum number of total cases that we don't need to change. So see, for the BRCA one, this structural variant data, so alteration amplification in 16.67%. That is the two cases, and this is for the BRCA1 gene. So here for the mutation amplification deletion in this study, so in the proteogenomic landscape of breast cancer of the CPTAC paper, they have mentioned about it. Third, in this study they have mentioned about the structure. So here they are also mentioned about plus 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 means uh, what alterations are there. Okay, not profiled. This is not profiled. They have profiled the structural variant data, they have profiled the mutation data, they have profiled the CNA data, or they haven't profiled, they have profiled only the mutation and the CNA data. Similarly here also no structural variant data, that's why they are you can get the information about only these two. So which study and what are the samples number this. So this is the, you can get an idea of the BRCA one. So you can mention it somewhere or write in somewhere that this is the cases in BRCA one. Now let us look at IDH one, which is known for the brain tumor. So you can see that in many studies there is no information about this. In this just 0.142%. Okay, here you can see that 0.41%, 0.09%, 0.37%, 0.15%. So now you all can understand, right, that how but mm, though it is seen that this IDH1 is significantly been reduced in the variant and the mutation data in the breast cancer, which is expected to be less in this data in this type of cancer, which is showing the trend. But if you look at it, still 0.55% or 1% cases are there where mutation amplification is there, in deep deletion amplification is there. Now you all can think that though there is this mutation, still this has not caused the brain tumor, but it has been then the question, there are two questions: is it involved in this breast cancer, what has happened? Or secondly, if not, then how this has been dysregulated, this mutation has been tackled by the body, but the mutations in the BRCA1 and BRCA2 could not be, and has caused the breast tumor. So am I clear? So what questions that we can arise? So that's why I gave this three data, these three comparisons to identify, to look at this, so that you can think about it, that where uh, how relevant is one mutation. And if, see, firstly the things what happen is that in a discovery phase study happens, where all the mutations, all the proteins, all the genes which are getting dysregulated are known. So I personally believe is that that work has been done at to some extent, because you can see that this many number of samples, so this many, if you see that getting clinical samples is very much difficult, and so many samples data has been analyzed across the globe of different kinds of population, all the data has been there. So we have known some markers like BRCA1 marker, BRCA2 marker, IDH1 marker for meningioma, NF2 mutations. So we have known, but now the next question would be that what are those statuses of those mutations in other cancers, other tumors? Then if that mutation is present in other disease patients as well, and that is salvaged by the body in that patient, then why in the different patient it is not being able to? Where are the changes happening? Where is the fault coming? And how we can tackle those, and where we can target novel drugs, or we can target those genes, or targeted therapy, you all might know, so that how we can treat those patients, or we can prevent those diseases. So that's why I wanted to show you this data. Hopefully you all got an idea. So so then is so so another thing is that you can look at the cancer type, but here the cancer type is that uh, only the breast cancer variants, so these two types of, so it's not an issue. So cancer study will give us, has given us much more clarity and information of these three genes. Next is the mutual exclusivity. This analysis, you can read that tested three pairs between three tags, means mutual co-occurrence and significant only. Okay, so BRCA1, BRCA2, neither present, A present not B, B present not A, okay, and both present, and the p value q value tendency is co-occurrence. So BRCA1 and BRCA2, if you see that the tendency is co-occurrence in a significant way. Okay, the p value is 0.003, much lesser than 0.05. Okay, and the log 2 and the q value, adjusted p value, is also less. So tendency is co-occurrence of the BRCA2 and the BRCA1. Okay, now if you go for the BRCA2 and IDH1, there you can see that the co-occurrence is 0.03. This is also showing significance, uh, uh, but not to that extent where BRCA1 and BRCA2 are present. Similarly for BRCA1 and IDH1. So now again you all can understand. So now again uh, exact Chitra, exact definition of deep deletion, I'm not sure. I have to look at it. I don't know if it's long deletion or what, so I have to look at it about the deep deletion thing. I'll look at it and tell you the in the next session. So the thing is that that's IDH1 mutation. This you can see that BRCA1 and BRCA2 are definitely linked to breast cancer. Definitely they are co-occurring, but in some cases BRCA2 and IDH1 is also getting co-occurrence is happening at some level, at some samples, in some patients. So why this happening? So might be, it might be, I am not telling, but it might be that the patient having breast cancer who is having both BRCA2 and mutation and IDH1 mutation, and we people are treating with people are treating with the BRCA2 level, but because of that IDH1 mutation, role of that mutation, that patient is not getting cured, and instead if that patient can be treated for the IDH1 gene, might be that patient will get cured, or the recurrence of the tumor will not come, the recurrence of the tumor won't happen. Or might be you can see that sometimes targeted therapy or radiotherapy, this also chemotherapy recurrence occurs frequently. So might be because of the role of those cancer, we don't know. So might be we need to think of the, so I think I don't know if you all are aware that now the personalized medicine, personalized uh, precision medicine is very much in the uh, hot topic for the researchers around the world. So where researchers have been compelled to think that the drugs that we are giving for in general, is it being effective for every person, or we need to think of go for the genomic profiling, or rather proteogenomic profiling, and identify the particular target for that particular patient. Might be the cancer might be the same in a whole umbrella, the breast cancer, but what are those particular mutations, or what are those changes at the phenotypic and the genotypic level, where we need to target those and try to treat those diseases. That's why I wanted to show you all that how we can think. So this will help us in thinking in a different way, that why this is happening. So this marker is also present in this. So what is this role in this marker? Are they, how are they regulated in a pathway? What are the protein levels or the metabolic levels? What are those changes they are making? If IDH1 is there, is it becoming more aggressive? Another question is that, for example, if breast tumor patient both are there BRCA1 mutation, but if one person is having IDH1 mutation, is it relating to more aggressive? It is relating to more uh, recurrence, more chemo resistant. So what are the roles? So that's why now it's the time we need to go ahead and look at the data very carefully and try to identify and interpret and see if we can gain some new information. If we go for the plots, so plots, you can see that there are various. So mRNA, so in this uh, for sorry, so in the so the BRCA1 and BRCA2 are known mutations, Chitra. So for the breast cancer marker, and IDH1 is a known marker for glioma. So if you go and search these three genes, so you will know that these mutations are major causes for the breast cancer. It is well-known marker. It has become well-known. That's why I took this three as an example, where IDH1 is a different in a brain tumor happening, and in the breast cancer happening BRCA1 and BRCA2. That's why I took these three examples. Yeah. So the firstly, what I wanted to say is that firstly we studied about the CNA versus mRNA correlation. So now if you generate a data where if only raw data is available, so just now appreciate the useful of these resources also, that if only a raw data is given to you, to go at that level CRNA RNA versus mRNA plot, it will take you enough time. It will not it will it require some expertise, it will require your time, it will require computational power, then only you can go. But because of this tool, now you can just go and click and you can get the plot for each study, that what is the CNA mRNA, and then you get full time to try to understand the biology behind it, try to understand that what, how interpretation can be done. Yeah, so here we can see that this is the deletion, deep deletion, shallow deletion. Okay, I'll try to look at the definition in the next class in a more better way, the what does it mean. So color samples by BRCA1 you can do, BRCA2 and IDH1. Another thing they have given of the clinical attributes: adjuvant, chemotherapy, treatment, diagnosis, age, cancer type. So you all can understand all these things by looking at this. Okay. So here is the mutation type, structural variant, and the copy number. Okay. So different. So data type we will use is the copy number, and we will use the log2 copy number values. Okay. So BRCA1 data type we will go for mutation. So you need to play with all these parameters to look. Okay. And the mutation and group mutations by mutated or wild type. Mutated versus wild type. Okay. So these you can see that there are mutations. So then BRCA1, what are those different mutations? Splice mutations, truncating mutations, inframe mutations, visceral mutations, and these are the samples with no mutation. Okay, in these samples. So these deletions and all are there, but no mutation is there. So these are the samples that are being uh, mutated. Okay. So you can change. So showing 122 with data in both profiles. So these are the data that have not been profiled with the copy numbers. So you can similarly mRNA versus cRNA, CNA correlation, protein versus mRNA correlation, Spearman correlation, Pearson correlation, what are the R square. So in this way you can go ahead and search and try to identify different data sets, try to interpret different data sets. Okay. So now moving on. So what other info? So I think you all can understand that the huge amount of information can be learned. There is a mutations. So if you go ahead and search in the mutations, so you will get everything. See the protein change, amino acid change, annotation, mutation type. I don't know if you all remember in the previous session where I had shown that from the SNVs or SNPs SNPs we can try to identify those, that what is the mutation type, what are those effects on the significance level. The missense, this is nonsense, this is splice. So for the particular study, okay, for the sample ID for each sample, so just think that how much information you can get. What is the cancer type, what is the protein change, what has been the annotation, okay, it is present in the cosmic database or not, what is the frequency, and this its type. So you can go and know about each and everything. See for IDH1 also you will get that in this, for example in this sample you have got this, and this is the protein change, right, in the sample, and this is the mutation type, missense mutation. So you can go ahead and correlate with the uh, for this sample what has been the clinical details of those patients, what was the age, is age playing a factor in for a particular disease, is gender playing, is hormone levels playing a different disease, is a type of treatment playing a different disease. So there are many questions, they are still unanswered and can be answered through different analysis and those. Next is the comparison survival. That another thing is that altered group, unaltered group, patients that selected group, patient level analysis in other labs. So here these patients are excluded. So so if you go for the BRCA1 here, you can go for the BRCA1. You can go for the survival. Okay. So here you can see that what probability of overall survival months. So altered group number of cases is 601, number of median months of the survival is this much, and for the BRCA1, so I think you all understood that BRCA1 altered, the patients where BRCA1 has been altered, what are the median months of overall survival, where BRCA1 is not altered, what is the median months of survival. So you can, from this type of genes, you can see every study doesn't perform a survival test. So from here you can understand that the mutation in a particular gene is being aggressive or not. If it is the overall survival rate is decreasing, that means this mutation is leading to changes in the genetic level, and it's creating aggressiveness tumor. So what are those significance, so you can understand its significance in more. So if you see IDH1, what is the thing, so you just change it, yeah. So just remove the BRCA one. So we'll just check with IDH1 what is happening. So IDH1, the median months is 144.67 months. So we need to check it that it has been decreased or increased. So if it is increased, if it happens that for a certain mutation in this gene it is increasing, that means it is leading to not so much aggressiveness. So what is the thing? So which study has done and how we can interpret. Then the copy number segments. So this portal is huge and it contains all information. So see here you can see that the copy number segment. So this is an tool IGV integrated genome viewer which has been integrated here. So here after you analyze your data, after you align with your reference genome, you see here it has been used as HG19. So I told you that the reference genome that is being aligned is generally the HG19 or HG. Now recent one has been HG38 which has come. So if you go by, you can go I think chromosome wise also the different segments. So in this IGB tool you can visualize your data. Generally it is being used to visualize your data. Similarly for BRCA2, IDH1. So if you all get familiar with these tools, you will be able to do it on your own or by aligning your reference genome. But it takes much time to come to this point where you can get automatically this data. Then the different pathways. Okay, so pathway name. So you can go ahead and check which are the pathways that have been affected with the mutations or the genes that you have queried. So I think everyone is clear with all this, or do you have any question? So I will suggest you go to this portal and try with the different genes, whatever you know. Try to go and identify, try to see what are those mutations, try to identify some prominent mutation markers just like I told you. So IDH1, NF2, BRCA1, BRCA2, also try to identify for these other cancers, and try to look that how they are behaving in different tumors, what has been the overall survival rate, has that been increased, has that been decreased in different, so many studies are there. So you all, if you go and start exploring, you will also get to know, get familiarized with the how to interpret those data, but most importantly that you will start thinking that why this is happening, why this mutation, uh, is it increasing the survival rate and decreasing, or what is the rationale, or can we target this change. So you will start thinking in this aspect, and also start appreciating the proteogenomics approach, why it is required at this point of time to characterize and profile these diseases. So this is uh, so so this is for all for CBI portal. Now another thing, as some of you have tried SRA toolkit, so I will request you, if you can use some virtual machine and all, just read. I am saying that start reading and start trying to do, because then the data that you are seeing there, you can also do it on your own, start analyzing, and you will also feel, means you can understand the aspect behind those, and will also help you in analyzing the data, in looking the data, and also thinking of the experimental designs and all. So we will just start one day. I will definitely show the different tools, and with some test data set. We cannot perform all those alignment and all because it requires time, and we but the results like this fast QC is another tool. Okay, so here we need to, when we sequence the data, so again the thing comes back, this we have gone with the analysis and all interpretation, but again it comes back to the technologies where you need a data which is very much robust, which is of very much high quality data. Then only you can carry forward and can interpret and analyze those data. So here you need to, when a genome sequence is being sequenced, so you need to see that how, what is the quality of the data. Is this data at the quality level? We can go ahead with this further analysis. So this tool helps us to understand what is the score. So like Q30 score cutoff is generally taken, which means that only one around 99, means all the sequences that have been reported here are not by chance. but actually that sequence is present here. So once the sequences get past those values, we take it forward for further analysis. So here also, so I had told that I'll try to give at least the names of those tools, and the step wise, that how the things can be gone. So SRA toolkit is used when you don't have your own data and try to get from the repository and try to analyze on your own. So the first step, the be it your repository data or be it your own data, you need to have a quality check on the data, and this is done by the fast QC tool, which helps you, gives this kind of report, generates this kind of report, and where you can go and then decide upon that you need to go ahead and further analyze or not. So start, try to read about all these tools and all, so that you will get both knowledge regarding that why it is important and how these things and analysis are done. So this is all for today. So any question, does anyone have any question? So you downloaded the SRA toolkit, right? It needs to be extracted, and then it needs to be path, it needs to be get provided, and once you provide the path, you can use the SRA toolkit. Then you need to provide a command and the SRA number. From there, it will start automatically downloading the data for you. So this is a command for uh, fast queue. One second, I just this is the command fast queue dump and split files. This is the command with the SRA number you need to give. After you provide this command and write the SRA number, it will start downloading the data for you. So if after the data is downloaded, the next step is to go ahead and for the QC file, for the QC check in using fast QC, HTQC. There are many tools, so fast QC is one of those tools to use. Okay, so any other question from anyone? All of you are clear, right? So about the CBio portal and today's session, uh, what has been discussed. Can I just yes? Okay. Any other one else or any other question from previous week or previous week assignment or anything? Week 1 live session lecture. Video, if you want the live session lecture, okay, so I think they did not post it the YouTube link, so I will ask for posting the YouTube link for both previous week as well as this week. Okay, so you can get so this session is completely recorded and is being streamed in YouTube also, so I'll ask to share those for the week 1 and the week 2 live session. uh, and any other question from week 1 or today's discussion? Okay, so if not, then I am closing today's session. Okay, so thank you everyone. So hopefully you will go and try to do the analysis in CBio portal and all. So okay, thank you everyone. I'm ending this session.